Kingsley. I'm Head of Researcher Engagement at UK Biobank. Thank you for choosing to spend your Saturday morning with us today. We've got a series of talks today from UK Biobank. I know many of you will be interested to hear what's happening with UK Biobank and what the future looks like. So I'll invite Klynn Alibocus to give a short talk with an update from UK Biobank. He's our Director of Engagement, responsible for how we interact with our participants, but also our research community as well. We'll also hear from Professor Martin Rutter, who is our Deputy Chief Scientist at UK Biobank. He's going to give us an overview of some of the omic data that's coming in, with a focus on what's happening with long read, and he'll touch a little bit on the proteomic data. We'll also then hear from Matthew Luce, who joins us from University of Nottingham today, with a really interesting talk about DNA methylation and that data that's coming to UK Biobank, and what that can mean for different disease areas as well. He'll touch on some of the work with nanopore and decode as well, so that'll be very interesting. And then finally, Daniel Seaton joins us from GSK, who's going to take us into the world of drug discovery and some of the insights that have already been discovered through the omic data within UK Biobank. We'll have time for questions at the end, and I'll invite the team to join a panel discussion as well. So if you have questions, we'll take those at the end. But otherwise, I'll invite Klynn to join and take the stage. Thank you.
Välkommen till Göteborg. Have I pronounced that right with my Swedish colleagues? Anyway, welcome to Gothenburg. As I said, my name is Klynn Alibocus. I'm the Director of Engagement. I've been at UK Biobank for about 5 months. It's been an interesting 5 months. But for those that are new to UK Biobank, who are we and what do we do and what is it that we offer? So why we feel that UK Biobank is special. For those that don't know, we're almost at our 20-year anniversary. So 500,000 men and women aged 40 to 69 years old since 2006 have given us their time, their blood, their sweat and their wee, etc., for available as research. It's quite a deep, extensive, so we've also done a number of lifestyle, environmental questionnaires with them. It's long. It's 20 years long and very deep. And coming from the healthcare industry, that longitudinal data is really important for health outcome research. And it's accessible. All of the data are readily available for any type of health-related research, and obviously bearing in mind the current incident, etc., which I'll go into a little bit later. But there are deep characteristics to participants which we feel that is special and unique to UK Biobank, and I won't go through all the list that's here. All I would say is our website is pretty comprehensive with everything that's on there, including some of the papers and outcomes that has been driven by UK Biobank research data. But I'd also say stand 800, that's where we are. If you want to come and talk to us and have an individual conversation with some of the lovely people from UK Biobank, etc., then please, I encourage you to come and have a conversation. If you're new to UK Biobank or want to know a little bit more about UK Biobank, do have a face-to-face conversation. We won't bite, I promise. I've touched on that there's a bit of an incident with UK Biobank, and I'm just going to take you through a timeline so that you get a better understanding, and we're sharing amongst friends here, of what's been happening and what is to come. So the timeline of events, and I would say there is a 55-page publicly available report which forensically goes through a high level of detail that was commissioned and requested by the UK government, etc. So it's freely available for everyone to come and see onto the website. Along with that comes with about 10 recommendations in the brief then. But we received a notification from a whistleblower that some of our UK Biobank data was up for sale. Right? That's definitely breach of our MTA. We did a series of investigations. We gave takedown notices. It was playing like whack-a-mole, trying to take stuff down, and it was coming back up. So we engaged with the UK government, who then engaged with the Chinese government, etc., and with Alibaba. The decision was made to suspend the wrap so that no other data could be taken out of that. I would classify that it wasn't a breach, it wasn't a leak, it was an infringement of our materials transfer agreement from some institutions from China. An ICO, which is the Information Commission Offices, which is our highest body in the country, in the UK, to look at data breaches and data integrity, etc. So they came and did an audit as well. On the 3rd of June, the CEO updated all participants, so all contacted participants were told personally what had happened, etc. The Oversight Committee, working with the UK government, produced this full and forensic report. We then had the participant webinars were announced so they can have direct conversations with the CEO, etc. And in late June, so for those who are currently working on UK Biobank, or existing research in UK Biobank, there will be a notification towards the later June explaining a little bit more detail and clearer around what's going to happen next. But let me be really clear on this one. The conditions of us reopening are really clear and spelled out into the report that you can find. And those are, we have to protect the participants' data. In that sense, there will be no downloading of UK participant data in the RAP platform. With that in mind, we also understand that the challenges that researchers have, that you can't separate out the analysis results from participant data, etc. And that's why we're building the world's first airlock or output checking service. So once those are better formed, better in place, and I can share that the procurement process is completed around identifying vendors that can help us do that. And that will likely be in the announcement towards late June, but I'm not privy to say anything more than that for reasons I've explained earlier. However, what can we all do in the meantime while we are waiting for the RAP to reopen? I would say these things, which again are in the report. One is, if you're an existing researcher and your project has expired or is out of contract and you have not confirmed that data is deleted via the AMS data deletion form, do it and do it now, okay? Just say that will be a condition of coming back into the RAP2, right? You've got to confirm that if you've had an analysis done or a project there and it's expired, confirm deletion of that data. Complete all the four mandatory. Now, you may have seen for existing researchers there was a flurry of activity in March and April around asking you to complete the mandatory training. This is not connected to the incident, but it's still important. We had about 80% of all of our researchers compliant, so there's 20% left that aren't. They need to complete all four, and these are listed for, and you can get access to those again via our website. And I would say keep an eye on what's going on with researchers like yourselves via our communities for news and updates and ongoing changes and development within the UK Biobank.
So I'm going to be talking about genetic insights that have been gained and will be gained hopefully through UK Biobank. I'm going to have a look back first of all at what we've achieved and then look forward. But first, just to say a few words about UK Biobank. This slide's been covered already. So Biobank is large, 500,000 participants, deeply phenotyped. We've got 20 years of follow-up in some individuals now. And the key thing, I think, is that the data's been made accessible in ways that hadn't been prior to Biobank coming on the scene. So it's really democratized data access. This shows a range of data that we have available in Biobank. So we've got lots of demographic and lifestyle information. We've got the largest imaging study in the world embedded within UK Biobank. We've got multi-omic data that we'll talk about in some detail, actigraphy in 100,000. And critically, all of this information is linked to health outcomes that have been collected through hospital records, cancer registries, etc., over the years of follow-up. So very exciting combination of data. It's the linkage between these data sets that makes Biobank really very unique. The data's been used by over 20,000 researchers in 120 countries. There's over 8,000 institutions using UK Biobank worldwide. And as you can see, the numbers are going up every year. And we've now generated more than 20,000 peer-reviewed publications. There's over 500,000 citations, and you can see there's no sign that that's dropping off in terms of the rate of output. So it's a resource that's been used internationally to great effect. So just looking back at what we've achieved in the genetic space over the years, the genotyping data, when it was released in 2017, probably put UK Biobank on the map for many researchers, and Since then, we've had whole genome sequencing data released in 2021, in 2023, whole exome sequencing in 21, and we've had metabolite and proteomics data, which we'll talk about later. So what does the genome-wide association study data, the array data, allowed us to do? Well, we've got GWAS data on hundreds of common diseases now and traits. Prior to UK Biobank coming on the scene, the GWAS data internationally was sometimes limited in statistical power, so we've changed that significantly. It's enabled causal loci to be identified in many cases, the development of polygenic risk scores, investigation of gene environmental effects, etc. So, and that integration with other data that we have in UK Biobank has really made the contribution of Biobank quite special. And as I mentioned, the open access has really made a huge difference to researchers around the world. I thought this paper was really quite important. So new methodology has been developed that will enable us to get insights into biology in the non-European participants of UK Biobank. So in this study, 7,000 traits were assessed and 14,000 additional loci were identified, and many of those were in those non-European participants. The figure shows that the common alleles in those non-European participants were often more significant than in the Europeans. So really great advances there, and it'd be great to see that methodology replicated in other studies. We're always interested in the clinical translation of genetic data, and there's been many polygenic risk scores that have been developed using UK Biobank data. The first was published in 2009, a very influential paper on cardiovascular disease showed that individuals in the top 3% of the risk score distribution were at similar risk to those with monogenic disorders in relation to cardiovascular disease. I think that really helped to show the potential benefits of these polygenic risk scores. In that same study, 8% of the cohort had a threefold higher risk than the background population, and the area under the curve for discriminating events was quite reasonable at 0.81. And there's been polygenic risk scores have now been used in many situations. They're being used routinely in many private practices, and we're starting to see some use in the NHS in England. So again, very exciting. The exome sequencing data has shown huge diversity in protein coding variation across the genome. We've got insights into function through the loss of function variants. And so these data have been particularly useful for pharma companies identifying drug targets. So rare variant associations with the trait in genes close to GWAS hits can help to give insights into biology and identify the relevant biological processes related to that GWAS hit. And finally, the whole genome sequencing data that was released has given us real insight into structural variation across the genome, and particularly the diversity that we see in relation to ethnicity. So we've got, in the figure there, we can see that the numbers of SNPs and indels and other structural variants related to ancestry here. So another major advantage for UK Biobank. So that's just a look back. I'd like to just start to look forward to the sorts of data that we might start to see in UK Biobank over the next few years. We have long-read sequencing that's going to provide us with some epigenetic data. That's in 50,000 using ONT. We've got array data from DNA methylation array in 60,000. That study has just been approved by our access committee, and that'll be starting just next year. We've already generated single-cell RNA sequencing data in 5,000 in collaboration with the Sanger, and we'd hope to make those data available to researchers at the next data release. And we also have infectious diseases data, so that's IgG data on 20 infections in 50,000 participants. We've now received those data and we're hoping to make those available to the research community as soon as we can. What are the benefits of all these new data sets? Well, Matt is going to probably tell us quite a bit more about long-read sequencing, so I'm not going to say too much about this, but we'll get, I think, huge insights into how structural variation is related to health and disease. Some of these structural variants may well have quite large biological effects. We'll have direct epigenetic profiling from the long-read sequencing that we get. We'll be able to resolve complex genomic regions such as the HLA locus and immunoglobulin loci, and so on. So huge benefits from the long-read sequencing that we'd hope to have released to our researchers at the next data release. And as I said, Matt will be saying a bit more about this later. Novo Nordisk have funded this study of DNA methylation using array, and this is going to be in 60,000 participants. We'd expect this will give us new insights again into disease prediction, help us to understand better regulatory mechanisms and causal pathways. We'll understand perhaps better how the environment influences disease through epigenetics. We'll get insights into aging through epigenetic clocks, and so on. So the 60,000 participants in this data set will be, I think, one of the world's largest DNA methylation data sets, and we expect that we'll be looking to expand this if we can get additional funding to support that work. The proteomics data in UK Biobank has been used by many pharmaceutical companies. We've got a talk later from Dan about GSK's use of this. So back in 2020, about 13 pharmaceutical companies came together and they funded this project, which was in 50,000 UK Biobank participants using the O-link assay of around 3,000 proteins. And I'm grateful to Chris Whelan for providing this slide, but the impact of these data has been enormous, not just for pharmaceutical companies, but for science in general. 3 Nature papers, over 2,000 citations so far. The data generated over 14,000 protein QTLs, which are really invaluable when trying to help identify drug targets. And Chris estimated from work with the consortium that there have been at least 20 new drug development programs that have been initiated on the back of the proteomics data that's been released so far. So that's a wonderful real-world benefit from these data, and we'd expect that there'll be more to come. So based on the success of that pilot project, there's plans to expand the proteomics work to include the whole cohort, that's 500,000 samples. We're also going to include 100,000 samples from the imaging assessment, and the increased sample size will increase statistical power. It will give us the opportunity to look at rarer diseases. If we just look at the common diseases, we can see that this increase in the sample size will increase the number of cases by between 4- and 8-fold for things like lung cancer and pancreatic cancer, which gives us greater statistical power to do these analyses. The samples that are coming from the imaging project will give us unique insights into brain, cardiac, bone, and vascular disease that we wouldn't have had before. So there's 100,000 participants that provided biosamples at this imaging visit. So again, extremely exciting. I just want to highlight this study, which shows the integration of genetic data and proteomics. This is bread and butter for the pharma companies, but I'd just like to highlight it because it's a nice example of the benefits of integrating these data. This was using the 50,000 participants that were involved in that first proteomics study. 4,000 of these individuals had a diagnosis of asthma. And the authors of this work looked at genetically predicted protein levels using pQTLs and their causal relationship with asthma. They explored this using Mendelian randomization and co-localization. And lots of interesting biology, much of it novel, was uncovered through this work, and I won't have time to go through the details. But one of the key things that came out from this was that many of those biomarkers, many of those risk-associated proteins weren't actually differentially abundant in the cross-sectional epidemiological analysis. And it's showing, I think, that many of these disease biomarkers are downstream of disease, they're consequences of disease rather than causal, and it shows the strengths of using the genetic data when combined with the proteomic data to understand those causal proteins in the disease process. So I thought that was a really beautiful paper, and I'm sure we'll see many more of those in the future. Our efforts in the proteomic space are going to be expanded significantly. We've got 50,000 samples that are going to be looked at using another affinity-based assay, the Sumologic Illumina protein prep assay, which is going to cover around about 10,000 proteins. We've generated a lot of that data already, and would expect to release that at the next opportunity. We have a mass spec project, again in 50,000 samples, that's been funded and developed by Elliptica Bio. Again, those data are generated, and we'd hope to make those data available to the research community at the next opportunity. So really complementary approaches that I think are very exciting. UK Biobank is moving to a new facility in central Manchester. The storage facilities there will give us an opportunity of increasing the rate that we can respond to sample project requests. We'll perhaps have about fourfold higher capacity to deal with sample projects. So you can see many more sample projects coming forward from UK Biobank in the future. So the Biobank is evolving into a multimodal atlas of human biology, I would say, and disease. We'll see more and more integration of genomics with multi-omics to transform the biology, therapeutic discovery, prediction, and prevention. So these assays are going to be done at larger scale over the next few years. There'll be deeper phenotyping. We'd like to see, more work in diverse populations. I've shown those multi-ethnic studies already. There'll be more AI-enabled integration of these data, and I'd hope in the future federated global analysis across multiple biobanks with different populations. And I think the challenge is going to be more about, not about generating data, but more about how we integrate these data from different data sets to understand the biology and improve human health going forward. So thanks for your attention.
Thank you to Biobank for asking me to come and speak. I should be clear that this project is one that I am cheerleading for, and I am not necessarily running, but DNA methylation in the UK Biobank. If any of you, like me, were up at 8:30 and in the epigenome session this morning, you would have heard lots of interesting information about what can be achieved by looking at methylation and what the diagnostic potential is for looking at methylation across patient cohorts. And I've taken this slide from Läufer et al. In Nature, which just shows the huge array of different information that you from looking at methylation across different tissues. And we already know that we can use DNA methylation to look at cancer, to look at dementia, to look at other complex diseases. I have one small issue with this slide, which is here, where they just talk about methylated cytosine. I am very passionate that cytosine is not just methylated. There is 5-MC, 5-HMC, and who knows what else that may be happening. But we know that we can do this, and we're already seeing it used clinically in many different ways. This slide is a non-exhaustive list of all the methods for detecting methylation. If you go and walk around the vendor area, I can guarantee there'll be at least 2 ways of detecting methylation that I've never seen there, if not more. But we're all familiar with the EPIC array. which enabled us to look at a number of CPG sites and has probably been to date the single largest driver of understanding methylation in the human genome. But that's looking at a limited number of sites. We've then seen the emergence of technology that allows us to look across whole genomes, from whole genome bisulfite sequencing to Illumina's latest 5-base method. And then we have the single molecule sequencing approaches, so largely PacBio and ONT, both of which can distinguish methylation while sequencing incredibly long molecules. And so I have, for a number of years, been very passionate about seeing long-read sequencing in the UK Biobank cohort, and in particular methylation. Now, we have been using methylation for some time. Sorry, this slide is, the title is slightly knocked out there, but we've been using it to look at CNS tumours, and this is possibly one of the most developed areas of using methylation clinically. On the left here, you can see a UMAP of all these different tumour types classified solely on their basis of methylation. And this allows us to do rapid diagnosis actually in clinical environments to distinguish different tumour types. And we know that these methods are going to be incredibly informative as we can look further through patient samples. This is just to show that actually we can do this comparatively. So there is some concern if we switch to different methods, we may not be able to integrate between them. So I am showing here most of the points are derived from epic array data. There are two samples that you can probably barely see at the top left there called new sample 1 and 2. What's the difference between those? One is a nanopore derived sample, the other is an Illumina 5-base derived sample, and we are co-clustering this data into a background of 2,900 epic array samples. So we know that the methylation measurements we get from these different technologies can be integrated together. So the exciting thing then is to see what else we might learn. Here is something that I've taken from a paper published in 2018 looking at DNA methylation clocks in aging. Being able to look at methylation over time and how it changes and use that to make predictions about changes in gene expression, about changes in phenotype, potential disease biomarkers. All of these things are possible. We have not to date had a large data set that we can look through. And so it's incredible to see what Oxford Nanopore and Decode will be doing in the UK Biobank data set going forwards. Where is this at? Well, I'm really grateful to ONT and Decode. They've shared some slides with me. The data are not yet available, but they are currently analysing these data, and they've shared some slides with me that I can use to highlight some of the things that they have been finding. So just to briefly touch on the data, there are 50,000 samples that are being generated, and the full data release is expected in 2027. The samples have been picked to cover a number of different areas. So the sample selection includes as many trios and duos as was possible to obtain. The sample selection covers longitudinal data sets as well, so there are participants who've been sampled at multiple different time points. There is a focus on metabolic and nervous system disorders where that information was available, and also cancer diagnosis. So the panel of participants is quite broad. There is initial research and analysis being kicked off by Decode and ONT, and they are looking to help drive a analysis consortia from outside of those organisations as well. And if you're interested in learning more about that, you can go and talk. I think ONT are at booth 310, but they've got a big circle thing above them out there. You'll see it. And they've also provided an email address for people who'd like to learn more, so ukbiobank50k@nanoporetech.com. So if you want to get involved in the analysis, please do contact them. What we know is obviously there are significant methylation changes, but from these data we're also going to get all of those large structural variants that have been talked about that might be incredibly useful for understanding basis to disease, basis to different pathways, and also potentially uncover novel drug targets. So how are the data actually looking? Well, the early results are looking very promising. They are able to generate 30x average mean chromosomal depth across all of the samples. So you can see in the top left here the mean coverage depth, and that has quite a pleasing long tail. The mean read quality is looking good, and the read length N50s are between 15 and 20 kb and are pretty consistent across the entire data set. I was involved back in 2018 using UK Biobank participant sample data to generate the first 7 long read samples. And I have to say those N50s are pretty consistent even now for those samples that were collected some time ago. The read length N10s are quite promising as well here. And so if you look across the data set that's been generated and you just top slice the data to the longest 30 kb of reads, on average there's about 2.5x coverage per individual that has been sequenced. And you can see the breadth of coverage across the genome in long reads as well. So yes, whilst the N50 is 15 to 20 kb, there's plenty of information here that's going to be incredibly useful for phasing and haplotyping going forwards. In the initial plan for this project, I'm aware they were going to do 5,000 genomes at this 30x coverage and then 45,000 genomes at lower coverage. Given the performance they've seen to date, I hope that we see an improvement on the coverage over those 45,000 samples as well. What's interesting then is to go into these data and have a look at what we find as we look at the harder to see regions of the genome, the so-called dark regions of the genome. And you can see in the bottom left here the mean coverage for the samples generated so far across exons, protein coding regions, tandem repeats and so on. And then you can see these dark regions and you can see that they are typically overrepresented. Why is that the case? Well, that's because those regions are often collapsed in the reference with respect to those individuals. So if you've got large segmental duplications or things like satellites in centromeres, they have been collapsed down, and so we see that in the coverage data as this amplification. And so it looks like the long-read sequencing is doing exactly as we expect, and we really look forward to seeing those data in their fullness when they're available. So as I said, all of these data should be available in the RAP in early 2027, and there'll be this consortia where we can look at it. As was mentioned, there is this orthogonal project going ahead, and this is being led by Jonathan Mill from the University of Exeter, and there are these 60,000 array samples being generated. Now, as I understand it, those are going to focus on longitudinal samples, and we think simply because of the nature of the sampling, there will be overlap with the existing ONT data. And so it's going to be really interesting to look and be able to infer and make those comparisons as well. I think this is incredibly important, actually, because we have a whole host of methylation data derived from arrays over time, and to be able to integrate that with the long-read methylation data as we go forwards will be incredibly useful. Thanks. I'm just going to remind you again of where you can get more information about this, and encourage people who want to work on that or look at the methylation data to register their interest by emailing that UK Biobank 50k@nanoportech.com email so that you can be signed up and contacted as things progress. Thanks very much for your attention.
Hi everyone, thanks for joining this morning, and thanks to the organisers for inviting me to come and speak. So I'm here to give a bit of a pharmaceutical company perspective on the UK Biobank data set, and especially the proteomics data, which we've been making very heavy use of, and actually looking ahead a bit to why we're excited to get more proteomics data measured in more individuals, more proteins measured across these different technologies. And as you can see, I'm especially representing GSK, but a lot of what I'm talking about is going to be very general for pharmaceutical discovery. So I'm going to touch on two things, really focusing on the biomarker discovery side of this. So in general, as a pharmaceutical company, drug discovery, we want to make decisions as early as possible in the drug development process. So if we can figure out as early as possible whether it makes sense to recruit patients, possibly thousands of patients, into a phase 3 clinical trial for a drug that might not work, we want to know that in advance, right? So we can avoid making these very, very expensive decisions late in clinical development. And so at the earliest stage, we want to develop a drug. We want to do some chemistry to target a particular protein. We want to engage in this target discovery step. And we have this very well-established set of methodologies for doing this now from genetics, where we can find a genetic variant that replicates the effect of what a drug will do in the patient and in treating the disease. And if we think about the UK Biobank proteomics data in that context, in diseases especially like cardiovascular disease and many immune diseases, as Martin talked about, if the relevant proteins are there in the blood, perhaps playing their causal role in affecting the disease. So, for example, PCSK9 in affecting the level of cholesterol in the blood, then we can, the blood is giving us a direct view of the disease-causing process, and we measure all those proteins, and we can make really clear inferences on which proteins we should be targeting, how much do we need to perturb them in order to have a disease-modulating response. So that's the target discovery side of it. You have to be quite lucky there, right? So you have to be working on a disease that is causally mediated by what's in the blood, like cardiovascular disease, and your particular protein of interest has to be there and has to be measured on the proteomics panel. So that's a bit of a special case. The more general case, so for all of the different diseases that we want to treat, is biomarker discovery. So here we're not actually concerned about inferring the causal actor on disease. Say we already know we have a drug target that is driving disease. We validated that from human genetics. But it's expressed in some other cell type, in some other tissue. It's not actually doing anything in the blood. Well, it's still important for us to know early on in the clinic that our small molecule or our antibody is correctly targeting that protein. And so what we want to have is a biomarker. So something we can measure in the blood that it might be completely irrelevant to the disease, but it tells us what's happening in the cell with our protein target of interest. And that means that if we can measure that in a small handful of individuals very early in clinical development, we can get some idea of whether our drug is going to work when we go to a phase 3 clinical trial in many thousands of individuals. And so because this is the very general case that applies to lots of diseases, I'm going to focus a little bit more on that here. So I'm going to use an example. This is not a current example. This went into clinical development about 10 years ago. It's not a GSK target either, but it's a very well-studied, genetically validated target. So this is TYK2. It's a member of the JAK kinase family, and there's a very well-studied locus shown on the right-hand side here. So TYK2 carries a common missense variant, which is associated with all sorts of autoimmune diseases. So we have this genetic validation, and this is being carried through to the clinic now. So there are compounds for TYK2 inhibition that treat psoriasis and are postulated to work for some other autoimmune conditions as well. And so if I frame this, that's the kind of target identification side of it. If I then look at this as a biomarker discovery problem, the interesting thing about TYK2 is that it's expressed very broadly. It does a lot of different things in a lot of different cell types. And so having developed an inhibitor for this protein, what you really want to know for autoimmune disease is that your molecule is working in immune cells. It's reducing the activity of TYK2, to a meaningful degree. And so you want something that you can measure in the blood that gives you that confidence that your drug is working, it's chemically doing what it's supposed to be doing. And so if I look at the UK Biobank proteomics data here, so now we have GWASs of all of the different proteins that we have in the Olink panel. There's a number of different signals that's co-localized with the TYK2 disease signal with its missense variant. I'm showing one particular example here, which is a cytokine, CXCL10. So this matches actually what you see in preclinical models as well. So I'm showing on the top here some data from mouse. So you can get you see the same thing dosing mice and measuring the CXCL10 levels as you would predict you would see from the human protein GWAS data. And so you could imagine if we were starting development today, we would be able to validate this much more quickly just in silico, just by referencing the UK Biobank data right from the first moment in which we identify the genetic link for TYK2 and psoriasis, rather than doing it after we've done all this preclinical work in mouse models and maybe even in some early clinical phases. And so for this to work in general across a broad range of diseases and a broad range of biology, what we're looking for in the proteomics data is for it to reflect kind of a rich set of biology that we can dig into. And so here I'm showing one way of looking at this. So I think we saw one plot from Martin about the number of signals that have been detected in the UK Biobank PQTL data. And now I'm going to focus on the diverse range of biology that captures, right? So it's not capturing just one or two dimensions of biology and just really hammering it with 3,000 different ways of measuring it. You've got a lot of different dimensions to this. So I'm showing in this heat map, this is taking, I've got 90 of the proteins with the most signals on each of these axes, and each point represents a degree of shared signal between these GWASs. And you can see in the top there, so CPA1 and CTRC, these are two quite closely functionally related genes. These are enzymes secreted by the pancreas. They're closely functionally related. They share quite a few signals, but there's actually a lot of signals that they have that are completely independent. And so even with related proteins, they capture different bits of causal biology that are, in this case, are happening in the pancreas. And you can see in general in this plot, there's a lot of stuff that's just completely independent. And some proteins, I think there's one or two proteins here that actually have almost no overlap with any other protein in the UK Biobank, in this 3K panel from Olink. And if you go and annotate these, it's also not just 1,000 different dimensions of immune cell biology that we've got here. We've got, as I mentioned, the pancreatic enzymes. There's some kidney-specific stuff. There's skin, epithelial things that you actually, before looking at this, wouldn't have necessarily expected to be getting a very rich picture of in the proteomics data, but they're here. And we expect, as we keep on collecting or keep on sampling more proteins, so the 5K and Martin also mentioned the Somologic 9K panel, we expect to keep on discovering new bits of biology about a whole range of organ systems that are useful for biomarker discovery. The other dimension that's interesting is, this is all tens of thousands of individuals. We're going to go up to 500K with all of the whole genome sequence, with all the individuals for whom we already have the whole genome sequencing data as well. And so I think it's interesting, even in the tens of thousands of individuals that we have at the moment, we can do all of the burden testing, all the rare variant analyses, and we're already seeing a lot of the same biology that I've already talked about in the common variant GWAS. We're already seeing that reflected in the burden data. So here I'm showing a similar plot. It's a different measure, so this is the pi1 measure of signal sharing between the burden test results, so using loss of function and missense variance here at a 0.1 allele frequency. And you can see again here in the bottom panel, it's genes with a nominally significant burden test result in one protein, how much do you enrich for small p-values in the other protein? And you can see these same groups forming here. So this is, again, the pancreatic digestive enzymes. There's a couple of eosinophil marker genes, and then there's a set of genes that are expressed in the skin. And so we expect, as we keep on increasing the sample size in the protein, this is just going to become a richer and richer data set. And I think we've even been a bit surprised that the burden testing is working so well, even just with some tens of thousands of individuals. And so with that, I think I'll close and thank, obviously, the participants in the UK Biobank and the organisers for inviting me. And then these are some of the individuals. We work very heavily with the UK Biobank data in general and especially with the proteomics data. So thank you.